So since we're starting one minute early, he asked me if I wanted to wait until the actual starting time or if I could tell a joke. And we discussed whether it would be better him telling the joke or me. And it ended up being me who was elected. So I used to play the bass, I used to have long hair. So when the journalists ask you how do you why did you want to play bass, I explained to them that you go to one lesson, you learn ding, you go to the second one, you learn dang, and you don't even show up for the third one because you're hired already to play in a band. I wish it were like that in biotech. It's certainly not. But simplicity can be of value also in biotech. And if you want to remember one thing from this talk, here it comes. To make T cells kill cancer, tilt is the answer. Oncolytic viruses have graduated from a field where they thought that the main effect on tumor control was due to the replication of the virus. An oncolytic virus means a virus which does not replicate in normal cells but does replicate in tumor cells. But it turns out that actually the main effect is caused by the immune response to this phenomenon. And there's one product approved, and there may be some merits to it, or it might be a prototype. What I think the field is going towards to is we're going to be arming the viruses for more effective stimulation of the adaptive immune response. It's been recognized that a huge issue, with the blockbuster immunotherapeutics, the checkpoint antibodies, is the fact that they only work in hot tumors. So if we can convert those tumors from cold tumors to hot tumors, that might have some real value. And most patients in need of new treatment approaches have metastatic cancer, so your drug needs to work in a metastatic setting. So our company is fully focused on making T cells attack solid tumors. So this is based on human observations. So even though we're technically a preclinical stage company, uniquely our entire approach is based on human observations due to a patient treatment series that we had ongoing from 2007 to 2012. Our lead product is armed with TNF-alpha and IL-2, and this is because we took the patient observations from the patients back to the lab, optimized it. And we have a pretty unparalleled scientific record when it comes to different models of cancer in the lab. So I don't think any other company has 100% cure rate for all of the relevant tumor models. So models of anti-PD-1 therapy, anti-PD-L1, TCR-T therapy, CAR T cell therapy, TIL therapy. We can cure all of those mice. Sure, it's animals, but it's a pretty good starting off point for human translation, I would say. We're now entering the clinic. We've raised about 12.5 million euros, 40 million US, but we continue to raise funding to be able to do larger and more meaningful trials. There's at least 3 problems that have been identified in immuno-oncology. One is that checkpoint antibodies, they are great, we love them, but they only work actually, if we're honest, they only work in a minority of patients, don't they? From 10 to 15%, 10 to 50%, depending on the tumor type, and that's not really satisfying in my opinion. And it's increasingly known which tumors they work in. They work in the hot tumors where there's neoantigens, the total mutation burden is high, there's PD-L1 expression, but an oncolytic virus can change all that, and even a prototype oncolytic virus like, let's say, Immulogic by Amgen can change part of that, but that virus was never even designed to impact T cells. In TIL therapy, which is an older approach, works quite well in melanoma and maybe also some other tumor types. The problem is not so much efficacy, actually it yields efficacy in 50%, but at a high cost of toxicity because you need to use high-dose preconditioning chemo and post-conditioning IL-2. We can achieve superior efficacy results with less toxicity by using TILT 1, 2, 3. And then there's the hottest potato in the room, is the CAR T cell therapies, which are working great, but only in CD19 positive tumors, which is about 2% of cancer. They're not working at all in solid tumors. Why? Anybody? Anybody want to volunteer an answer? I can help you out. It's because of the immunosuppressivity of the tumor microenvironment, and we can change that. So the CAR Ts are effective against tumors if they have a chance to be effective. So our virus modifies the tumor microenvironment so that the CAR Ts can have efficacy. So unusually for a preclinical stage company, our approach is fully based on human observations. So we, from treatment of 290 patients, we realized that following oncolytic adenovirus treatment, the tumors become inflamed. The lymphocytes leave the blood and go to tumors, and that there's systemic transduction of tumors through the intravenous route. So it actually doesn't make even a difference if you inject the virus intravenously or intratumorally, because the tumor acts as an extended release device. So when the virus replicates in the tumor, the virus ends up in the blood and into distant tumors. And it's pretty cool. It uses cells of the blood for hitchhiking in the tumors. Can you imagine that? I mean, it uses cells as taxi cabs, basically. And this is not science fiction. We have the data for that. I can see many of you nodding in an excited manner, and you should be. It is really cool. So our virus is modified in 7 different ways. Might sound like a lot, but we spent 19 years honing our skills. So there are several safety modifications which restrict replication of the virus to the tumors. And I would say it's a pretty cool way that we've restricted the expression of the transgenes to tumor cells, so that only in cells where the virus replicates is the transgene expressed. And then we use a modification of the capsid to allow entry through desmoglein 2, which is a tumor-associated molecule instead of the usual Coxsackie adenovirus receptor, which is not necessarily a tumor-associated, and in fact the Coxsackie adenovirus receptor is downregulated in advanced tumors. With our optimal construct resulting from almost 20 years of work in the lab, we get pretty good efficacy in our lab models. So we often end up using a pretty exotic model, a hamster model, a Syrian hamster model, because in mice our virus does not replicate, but in Syrian hamsters, yes, it does. And we get pretty good efficacy, but we don't cure everything. But then when you add T cells we're able to cure 100% of the mice. And this is satisfying because the virus was designed to stimulate the adaptive arm of the immune response. And if you challenge those mice with the same cell line, they're completely protected from the same tumor. It's interesting that they are not fully protected from the same tumor when the treatment agent was not armed with a transgene. And also if you challenge the poor hamsters with a different tumor cell line, they are not protected at all. Checkpoint inhibitors, the transformation of immuno-oncology. I mean, I bet this meeting was like 2,000 people about 5 years ago, and now it's what, 15,000, 20,000 because of checkpoint inhibitors. But unfortunately, this is actually similar to what is seen in humans. So checkpoint inhibitors alone are not that effective. I mean, they're effective in some patients, but not effective in others. And it's known why, and I already told you that it's because of the neoantigens and stuff like that. But with our virus, which was designed to stimulate the adaptive immune system, we can get a lot better results. I mean, how do you like these figures? What do you think? If you were a patient, would you rather have a therapy which yields this sort of results or this sort of results? CAR-Ts are really, really promising, but they're still a prototype approach in the context of solid tumors. The problem is not in the specificity of the CAR-Ts. I mean, smart people like our collaborators at the University of Pennsylvania, they can make those CAR-Ts very specific for a certain target. But if that tumor is very immunosuppressive, the CAR-Ts are not going to be able to do their job. So it's really a match made in heaven that our virus modifies the tumor microenvironment there at the tumor. So the CAR-Ts are killing the tumor cells. Our approach is modifying the tumor microenvironment. And that's why there's 100% survival in these animals. And this may be a simplification for business people. If there's any VCs, just focus on this. There's more brown here because there's more trafficking of the T cells there to the tumor. But to be honest, if there's some scientists out there, you know, I have to be honest, it's a bit more complicated than that. But it does work, and I think that's how we'll cure cancer. So right now, Tilt as a company is fully focused on our first technology, Tilt 123, the virus-armed routine of alpha and IL-2. But we do have ambitions to move also our second generation technologies into patients. Although we were founded only in 2013, in a way our story starts a bit earlier because we had the patient treatment program which started in 2007. We treated 290 patients in an individualized treatment approach. And we're an extremely talented team. Maybe there's some Europeans in the audience who'll recognize some of these names. If you're Americans, you may not recognize all of them. And in summary, T cells are the key to efficacy in all forms of tumor immunotherapy. Tilt 123 is the only oncolytic virus specifically developed for recruiting and activating T cells. We have demonstrated 100% efficacy in animals treated with Tilt technology in the context of checkpoint inhibitors, CAR T cell technology, TCRs and TILs. In humans, we've seen systemic efficacy generated by intravenous transduction of metastases and systemic immune response. And to make sure that we don't forget what it's all about, let's see if this works out. This is the riskiest part of the presentation. This is from Finnish TV. So this is one of the patients in our Advanced Therapy X program, the individualized treatment approach. So this guy, Akseli, he had a neuroblastoma which was metastatic to his bone marrow. So he had to, he was not able to go to preschool because of the pain due to the bone marrow metastases. But 1 month after we had given him the oncolytic virus treatment, he was able to go back because the pains were gone, and he's here playing with his sister. So, at least for me, that's what it, the thing that makes it count. Thank you very much.